Hello, everyone, and welcome to Cell and Gene Live. Our topic is scaling cell therapies, auto versus allo, and I'm your host, Erin Harris. I'm joined by two of the best and brightest the sector has to offer to discuss our topic. Michael Blackton, SVP Cell Therapy Manufacturing at Nurix Therapeutics, and John Lee, VP Head of Cell Therapy at Center for Breakthrough Medicines. We're going to dive right into our discussion in just a second, but before we do, please note we will be taking audience questions. So submit your questions for Mike and John via the Q&A tool, and we will get to as many as we can. Also, I'll be asking you to respond to 2 poll questions throughout the event. So please take a moment to respond when prompted. All right, on to the good stuff. So if you're a repeat viewer of Cell and Gene Live events, you know I like to start out by setting the stage and level set the conversation around our topic. And so scale up of a process involves using larger vessels to increase the volume of production. In scale out, the culture vessel remains the same, but more units are added to a system to increase production capacity. And any discussion regarding cell and gene therapies inevitably includes the challenges of manufacturing them at scale. And so, John, I'd like to start with you. Broadly speaking, talk to us about where we are as a sector with autologous scale out.
Yeah, it's we're at a critical, almost an inflection point right now with regards to scaling out autologous therapies. We have almost a handful of FDA approved cell therapies, in the oncology space, as well as some others in non-oncology applications. And, we have another 1,500 or thereabouts clinical trials underway. So understanding what's going on with scaling out and the limitations of potential solutions is extremely important. Kite kind of leading the way there. Early on, Kite obviously had their manufacturing in California. They expanded into Europe, into the Netherlands, and more recently opened up another facility in Maryland. J&J obviously is doing similar things now as they ramp up to increase commercial production for Carvykti, their BCMA asset. As I see it, there are a couple primary challenges. One is around regulatory. The regulatory environment, obviously the FDA and European Medicine EMA they require as innovator companies and manufacturers begin to branch out and create their products in other venues, there needs to be a comparability assessment that's done. And this is a key thing that needs to occur as we're scaling out for those autologous products. Similarly, just from an operational perspective, unlike Allo, where scaling up is necessary, the scaling out really, it falls on a number of things. Obviously, you've got the facility, you have the equipment within the facility, as well as the personnel. And as a general rule, the world doesn't have enough qualified personnel at the moment to really scale out as effectively and as quickly as we would otherwise want. In terms of solutions, I think there's a couple ways that this can happen in autologous therapies. Reduced harvest manufacturing is an area that is gaining steam. Novartis last December, at ASH, they released or described their quote T-Charge platform. This is a manufacturing platform that allows for harvesting of autologous cell products in about 48 hours. And Gracell is another company that's also doing this. And what this enables then is really to maximize efficiency within a manufacturing facility. When vein-to-vein times were previously anywhere from 14 to 28 days, some of these new manufacturing, reduced harvest manufacturing processes are really bringing that in to just a few days, which allows then to really increase the efficiency of those same facilities. And then, of course, it goes without saying, but I'll say it anyhow, obviously integration of digital capabilities, manufacturing execution systems, electronic batch recording, automation, and so forth are other potential solutions, but these are more in their nascency at this point in time.
Yeah, good point. You bring up a lot of topics, even as you just talked there, that we're going to cover throughout the rest of our hour. But before we do, I want to make sure that we hear Mike's perspective on this. Mike, from your perspective, thoughts on what John just said, in addition to your take on the sector as from an autologous scale-up perspective, where are we?
I think from an autologous scale-up perspective, we're really looking at two things. One is, scale up or scale out within a facility, and that's where you get into cross-contamination issues and ensuring product safety within that facility. And there's various facility designs. There's, we probably heard the term ballroom concept, which presents some opportunities and challenges versus individual rooms per patient, for example. The other aspect is scaling out across geographies, because you're dealing with not only supply chain issues across different geographies, but also jurisdictional issues with different regulatory requirements. But I think it all comes down to life cycle practices. If you have adequately thought about your process and developed that process with consistency in mind, taking into account aseptic processing requirements, for example, cross-contamination, patient scheduling, then you're well suited to a scale-out scenario. Now, rather than talk about individual technologies, I tend to think of these in terms of broad brushstrokes, because every product is different. But remember, if you have an engineered autologous product versus, say, a TIL, there are different requirements. For an engineered product, we have vector control and supply chains versus the cell therapy control and supply chains. You really have kind of two products there. And how does that relate to your scale out scenario? So if you have one source of vector, for example, and multiple geographies where you have cell therapy processing, that presents a different challenge than if you had multiple starting material or vector manufacturing sites and multiple geographies where you're scaling out. So that's kind of the way I look at it. And the other thing I think is important with these, and I'm a real advocate of automation of these processes. Again, requires a lot of process capability and control. We have to think about the core requirement, and those core requirements are life cycle, post-approval changes, right? So we should always assume that we want to develop our product for future changes to make that happen in a expeditious way. And John talking about comparability is a good example of that. What are we comparing? How are we comparing it? What constitutes comparability? It doesn't have to be exactly the same, or is it substantially similar? What do these terms mean? It's all different for every product.
Sure, that makes sense. I want to have the exact same conversation, but from an Allo perspective. We have FDA approved autologous therapies, but where does the potential lie for allogeneic? John, let me ask you that. Let's just have the opposite conversation.
Yeah, with Allo, I think it's a bit earlier. We're still really trying to focus in on some of the rate limiters, which are largely include downstream processing, harvesting, formulation, those kinds of things. We're dealing with increased volume there from a scale up perspective. But there's an underlying issue with allogeneic, and that is really maintenance of the integrity of the drug product, as measured by functionality. You can imagine, there's a big push, especially in the world of T-cell immunotherapies, to adhere to this stem cell memory-like phenotype. And as you try to achieve the numbers of cells that we're talking about in the allogeneic world, a typical harvest, at least the goal is somewhere between 10 to the 11th and 10 to the 14th cells per batch. To achieve that level of expansion, the possibility exists that you're going to end up with a phenotype within that population that are more differentiated than ones that have not been expanded to such a degree. So finding that balance between expansion as well as functionality is a key component here. And then, downstream from that, those more logistical or operational constraints that I mentioned with regards to downstream processing, in some cases, 2,000 liters worth of material to process, filling that drug product on the back end and maintaining consistency from vial 1 to vial 500 it's a logistical constraint that folks are contending with at the moment. But I think, as we're discussing, it's a problem that is not completely solved. And, the field is getting there. We're seeing fillers with better capacity and mixers that will accommodate microcarrier-based cell culture. But at present, a one-size-fits-all solution to some of the problems that are facing the Allo scale-up are just not available as of today.
Yeah, you're not kidding. We talk about that on Cell and Gene all the time, how there's just several problems that have yet to even come up and to be figured out. Mike, I want to get your weigh in on this. Before we do, I'd like to put up our first poll question, which we will get to later in the hour. But Mike, what's your take on the Allo scale up? Where are we, and where does the potential lie?
So, my background is in monoclonal antibodies, and monoclonal antibodies back in 1995, 94, we thought these were not characterizable. We're never going to be able to make these large volumes. I mean, we were in very small bioreactors, hollow fiber systems, for example, adherent cultures. But today, monoclonals, it's not even CBER, it's CEDAR. It's very rote. I see that with allogeneic. The problem is, I think the technology is very much earlier than it was even in 1994 for monoclonals, where you have, for autologous, you have scale out. For allogeneic, of course, we have this opportunity for scale up. But what does that really mean? There are other issues, right? We have, for autologous, we have starting material variability. Each patient has different starting materials, so you have to address that. With allogeneic, we have cell banks to deal with, and there's some allogeneic products that have multiple cell banks that have to be managed and characterized, right? That's very similar to the starting material challenges we have with autologous, but the benefit is you have a consistent amount of starting material, and your cell banks are consistent. The other piece that you really don't have with autologous is this donor eligibility piece, which I know is not really germane directly to manufacturing, but that is a concern. And the genetic manipulations we do as we develop these allogeneic products could become a challenge because, of course, we don't know what all the oncogenes are. One of the concerns with developing stem cell allogeneic products, of course, is oncogenesis. And then the other piece I have on my notes here is, long process, right? So some allogeneic products, while you can scale up to, 2,000 liters or more, some of these processes can be months long and have multiple genetic manipulations and scale up issues kind of serially. But once you get that solved, it's very conducive to scale up. Long process. I'm worried about aseptic fitness, right? Like container closure integrity and process integrity. While we are more, conducive to traditional scale-up pathways, it really remains to be seen whether you could go beyond, 3 to 5 liters in a cell culture system. The other thing that becomes important with allogeneic is this notion of real-time process monitoring, which we've been struggling with in traditional cell culture for years. I think it becomes very important given the length of these processes and the aseptic issues. So if we can have, online cell counts and of course pH and all the other parameters, and be able to react to those, and characterize using those, that would be really important.
Good. Okay. And Mike, I'm really glad you brought up both starting materials and cell banks. We're going to get to both of those in more detail, specifically with starting material. When it comes to quality and consistency of raw materials, acquisition of those starting materials, it's obviously critical to the success of the therapy. Most cell therapies today are produced from a single patient at a time, using that patient's cells as the starting material. That one dose at a time approach can be difficult. And so what I'm asking both of you, what are the specific roadblocks for autologous when it comes to starting materials? Mike, I can start with you if you'd like.
I tell people in the industry, if we just look at it very basically, I'm making a salt solution, let's say sodium chloride and water. And the benefit of that is we know exactly what that sodium chloride is, right? NaCl, 99.9999% pure. What if it wasn't? What would we do? We'd have to make a lot of batches of salt solution to characterize it. It's the same thing with autologous therapies. Every patient is different. Tumor microenvironment different. If for TIL, the apheresis could be different for traditional like CAR T, and that presents a burden of characterization, which is why I'm a real proponent. We have to really think about this from the very, very beginning as we develop these processes, understanding that there will be post-approval changes. So how do you manage that from a regulatory perspective, right? It means we need to test a lot of patients. That's in a nutshell, the way I look at
it. Good. Okay. John, I want to get your perspective too on starting materials and maybe some of the specific roadblocks from an autologous perspective.
Yeah, I mean, I think dovetailing on what Mike has said, what effectively what he's describing is variability or heterogeneity of that starting product, right? The cells, across different patients as they're collected in apheresis, patient A may have seen in some cases 6 lines of prior for some of these clinical trials, and the T cell compartment there is effectively at the least compromised, and at the worst potentially destroyed. And so, trying to make an efficacious cell product from that shoddy starting material is in many cases difficult to do. And of course, we probably should have led with, from a supply chain perspective, both plasmid DNA and vector are both at present limiting with regards to gene modified cell therapies. These are both at present, in most cases, backlogged, and companies are waiting on them. There have been a few articles of late. Wall Street Journal, I know, published an article not long ago, basically that there are patients who are dying while waiting on their therapy simply because there's not enough manufacturing capacity worldwide to treat these patients. So the starting material then becomes extremely important if that's the rate limiter, and vector and pDNA are both part of that. But returning back to where we started, it really does come back to this heterogeneity and understanding, how to normalize that. And I think there's a couple potential ways to mitigate the risks that come with some of this variability. One is, and we're seeing this already, as these therapies are approved, they're moving into earlier lines. And so, the starting product then gets hit less with things like radiation and chemotherapy prior to creating cell therapies. There's the concept of at-risk banking. So when a patient comes in on day one and he or she is diagnosed with some sort of disease, at that point, that person could get an apheresis and then have that stored for the appropriate time to create a cell therapy. Now, that obviously creates a whole logistical, cold storage infrastructure question, which we won't get into here, but it's certainly an option. And then, lastly, and I think this is worth pointing out, there are companies out there at the moment that are retrospectively looking at data from patients who have done well on these therapies, and they're defining the characteristics that are prognostic or otherwise predictive of response. And that'll enable manufacturers then to start building those features into those products over the long term.
Okay. Those are, you hit upon two very good points. And the first one is that starting material in the clinic could be very different than what you see commercially. We're starting to see that as these approved cell therapy products are surveilled in the industry. The other one is this, as you said, I call it the holy grail of cell therapy, is to predict the outcome, right? This is where the role of translational comes in, and looking at biomarkers and things, and being able to really understand what characterizing that starting material means. It's also indicative of where we are technologically or scientifically in the industry, where we have a lot more growing to do. One note on scheduling for the autologous products, and given the fact that patient, we call vein to vein or arm to arm time, or whatever you want to call it, is very important. That relates to the scale out strategies, right? Where typically in a monoclonal facility, when I used to work in monoclonals, we were talking about 95% plus equipment utilization, meaning that equipment is making product 95% of the time, and 5% of the time is down on maintenance or other issues. For cell therapy, for autologous, because we have to respond to the needs of the patient and the scheduling, if we get above 50% equipment utilization, we're golden in a commercial environment. So so you have to really understand what that means for your organization. Does that mean, and this is where automation comes in, you have 25 operators sitting waiting for the patient, only 12 of them in use at any one time, or do we use automation to help with that equipment utilization issue? So those are some very good points. I would love to be, I wonder what it'll be 10 years from now with regard to starting material and how it can predict an outcome of a cell, or at least a cell culture process. It'd be interesting to see what'll happen.
Yeah, probably light years different from where we are now, I would think. I want to make sure we're having the same parallel conversation around, donor cells, and from a scale perspective, let's talk a little bit about, the allogeneic side of this, and from a potential, where does the potential lie, and what are the challenges? Let's talk from a donor perspective, same type of conversation. John, you can.
I'm sorry, I missed the question.
Oh, sure. Sorry. So from a donor cell perspective. Oh, okay. Sorry. Can you hear me now?
Yeah.
Okay, good. John, I'll start with you. From a donor cell, I want to make sure we're having a parallel conversation and talk about it from an allogeneic perspective. What are the biggest challenges when it comes to donor cells for allogeneic therapies? Where does the potential lie?
Yeah. It's a quite a contrasted conversation around allo when we're talking about donors, because typically there's some level of gene editing that needs to occur, and as a result of that gene editing, you've got, at least in my eyes, two primary concerns. One is around just safety, and we can talk about that in a moment. And then the other is the resultant scale that you can achieve as a result of whatever gene editing that has been done. Typically, without getting too much into the weeds with regards to the science, but the intent of the gene editing is to eliminate allogenicity, whether that be graft versus host disease or just general immunogenicity and rejection of the drug product. But from a safety perspective, after that gene editing is performed, we as manufacturers need to ensure that the genetic stability and otherwise the genetic integrity of the resultant cellular population is indeed safe, and we're not otherwise subjecting the patient to additional risk through genomic abnormalities. From a scale perspective, and I think I heard somebody else on your other podcast, the CEO of Poseida a few weeks ago, Aaron, he was talking about I forgot what he termed it as, but it's something that's very relevant, especially in the world of T cell therapies. We're knocking out the TCR to eliminate graft versus host disease, and how 99% of T cell therapies are expanded to scale is through engagement of that same TCR. So when you knock that out, there need to be other ways of expanding those products. And companies like Poseida have already developed these strategies, but it is one that may be suboptimal, at least compared to what we're currently doing with CD3 beads. And then I guess the other component is understanding, and I think I mentioned this at the top of the webinar, as we start to expand these cellular populations into the 10 to 11th, 10 to the 12th, there is the risk of functional exhaustion, and navigating how to get that expansion without compromising the functional integrity of that cellular population is an area that a lot of companies are looking into at the moment.
Gotcha. Okay. Before we move on to aseptic processing and that topic, I want to make sure, Mike, I'm giving you the opportunity to chime in as well on, from your perspective, the biggest challenges with regard to donor cells.
I mean, I think that John hit the major points. Now, my expertise is in manufacturing and not in designing these types of products. However, the TCR knockout issue is I think was something that was in my mind. Also, the genetic stability of your construct as you manipulate these cells, I think, is something that has to be investigated. How we perform those genetic manipulations, but also when we look at the donor, the typing of the genetic type of the donor cell itself, I think has a lot to say about success rates, but this is kind of beyond my expertise. I'm more of an aseptic processing guy.
Well, then this question's for you, because we're about to cover aseptic processing. All right, so I want to go through the issues that can affect the manufacturing process when it comes to aseptic processing. So perfect segue. Talk to us about, Mike, what are the critical elements to successful aseptic processing?
So, we had talked before, and I talk about this a lot in the industry. If we look at the beginning, right, we take an apheresis or a tumor sample, we apply it to our culture. And aseptic processing guidelines say anything added to an aseptic process has to be sterile or able to be filtered to be sterile. Well, you can't filter a living cell. And contrary to what this, I guess, is now in my opinion, you cannot take an apheresis sterilely. It's not sterile by definition, because taking that needle jab is just not sterile. But we treat it as aseptic and sterile, right? So beyond that, what I look at is any manipulation that can impact sterility has to be addressed. If we look at a typical TCR, I was involved with one process where we did a aseptic processing risk assessment. We looked at every place, every area in that process where we can get a contamination. This document was well over 500 pages. And one of the things we did, we look at tubing sets, reduce the number of manipulations tubing sets. So we started making custom tubing. And you could do one tubing set, and it could reduce 25 pages of that risk assessment and make it easier. There were some, in the early days, we had operators modifying commercially available tubing sets to fit the process. That right there is a risk, an aseptic risk. So we really have to think about how we execute this. The challenge in cell therapy in general is it's very academic, and it, working with CMOs that have a little more commercial experience or getting people from like my background in monoclonals can help us design these processes with these aseptic practices in mind. Now, there are differences jurisdictional differences between this. Although we are harmonized, I can tell you there are different countries have different concerns. But if we look at the process holistically from an aseptic perspective and use risk assessments and risk management methodology, then we can really target what the product needs to maintain sterility and safety. The other piece is our lot release. We talked about the need for rapidly providing these products to the patient in an autologous setting. This really has resulted in us exploring these rapid sterility techniques to be able to get a sterility readout as fast as possible. So an example, a challenge, I was talking to somebody at a very large monoclonal facility, and I asked this head of quality, how long does it take you to release a batch? Well, this also relates to allogeneic, I guess. Everybody, working on 6 to 8 weeks. Well, in autologous, we're talking about 6 to 8 days. Mm-hmm. So how do you get to the point in an aseptic process where you could release a product purported to be sterile within 8 days of the last manipulation? And the only way you could do that is with rapid sterility. Developing rapid sterility is a challenge as well. So I look at it in terms of really two things. What are the analytics required to ensure sterility in process? That includes in-process sampling, and then the manipulations that are required and those that can be removed from the process. So fewer manipulations means a safer product. And this is where automation comes in and everything else. So that's kind of my thoughts.
Good. I want to move on now to single-use solutions. We do have quite a bit to cover, so I want to make sure we're covering everything. The poll question we asked, and this will help us, deliver our risk and talk through the next topic. What's your biggest concern regarding using single-use solutions? And so it looks like at about not a great majority, but cell damage during processing seems to be the biggest concern. And from lab bench to bedside, disposable processes, supporting regulations, and all that can be vastly different from autologous and allogeneic therapies. So, one, do you agree with what the viewers have said, and do single-use, ready-to-use, disposable components and systems play a role in scale, and why is this? And John, I'd like to start with you for this question.
Yeah, I mean, looking at the poll results, I'm actually struck by I guess the evenly distributed nature of the responses, right? I mean, you've got effectively 1, 2, 3, 4, half of the responses are probably within the margin of error for the number of folks who have responded. Sterility, cell damage, particulates, and then the closed system processing all come out fairly high there. And it's somewhat not surprising because there's, this is an area that is still burgeoning around single-use solutions in the world of cell therapy. And we're getting to a point now where I think you're going to start seeing more and more investment into this space because of what it can do to scale, going back to where the question originated. The big advantage of disposables, obviously, is that the changeover turnover times are lessened simply because you can dispose of them at the ends. So that means less manual labor to clean. They're obviously pre-sterilized, so there's a reduced risk of cross-contamination, all of which results in increased efficiency within that suite. I think that's effectively, and I guess one more thing to say is that at least in the world of cell therapy at the moment, we're running into less issues here in terms of availability of disposables because in the world of vector, I think it's a bigger concern at the moment, simply because those folks are competing against vaccines in the midst of a global pandemic.
Yeah, sure. And Mike, you and I leading up to our event today, we had discussed this offline, And I wanted to get your thoughts on, regarding the single-use components. Do auto and allo differ? If so, what's your take, and even, your perspective on the responses here?
So it's funny, when we talked about this, my answer was all of the above. Mm-hmm. But on that particular issue, it's one thing, when we scale up a autologous product, we're talking about, going to 5 liters, okay, to get a higher dose for a particular patient. And there's technologies that are actually very scalable from 100 mils to 5 liters. I can think of a couple, right? For the larger bioreactor systems, though, I don't know how that scale up will work for an autologous or allogeneic T cell. We see systems up to 2,000 liters in literature for these types of products. My background in cell culture is, 10,000 liters scale. And we know that for a monoclonal, that's relatively scalable, right? I just don't think there's a lot of experience. So going back to this characterization issue, we need to really think about, and product definition and life cycle, we need to think about scaling as we develop these processes. Think about the end point. When I look at this, just coming back to this poll, most of this is about sterility. But this one piece, cell damage during processing, is a pretty large thing. This was something we talked about 5 years ago. And it was like, you don't have to worry about it. But you do. And I think that as you scale up for an allogeneic product, you may have, if you have larger piping, maybe you have less shear forces. Thinking about physics, maybe it scales better at large scale than small scale. Maybe you have a better culture. We saw this in chromatography back in the early 90s when I was in purification. It needs to be investigated, but I think that just saying we're going to scale up with single-use systems and just say it works, you got to develop it. I remember a time years ago, I spent 46 hours straight in a clean room because it was just a filter. It's never just a filter. Everything needs to be developed.
Mm-hmm. I want to move on to supply chain challenges. I feel like we can dedicate an entire hour just to this, but we know that it's about managing risk and having strong partners to work with to keep risk at a minimum. Speed to market is an important consideration, and we have to ensure that supply chains for the critical components can support product duration and mitigate potential delays. But John, I'd like to start with you. Talk to us about the differences in supply chain challenges that may hinder scalability, but yet provide existing solutions or maybe even best case scenarios for an autologous setting.
Yeah, this is an area of as we're building out capacity here at CBM, continues to be at the forefront of many of the things that we deal with on a daily basis, is really trying to understand how we can better forecast our needs against in some cases I think we're potentially in a worse case than say just an innovator company with defined products. So we're kind of the test case, or the crash test dummy, so to speak, with regards to understanding how supply chain considerations in the midst of a global pandemic affect scale. I can tell you a little bit about our experience, what we're doing internally, and bounce that off of Mike as well. The number one thing we're doing is really using data to drive forecasting, predict demand, so that we can ensure that we're thinking about things in the right way with the right capacity on our side, as well as ensuring that the supply is going to be there to accommodate that And so we have frequent, and when I say frequent, in many cases, daily S&OP discussions just to ensure that the left hand knows what the right hand is doing. Of course, a big part of this, and I think you've already mentioned this in the lead-in, Aaron, is having just top-notch supplier customer relationships. And one of the bigger problems that we've had personally is with certain suppliers changing lead times on us and then not informing. And so, the way to get past those issues is to have weekly, if possible, updates with them to understand the orders that have already been placed, as well as updating lead times associated with things that are longer expectation. And then another part of that is just to ensure from our ends and anybody who's in the world of manufacturing, really to try to ensure redundancy across your supplier matrix, right? If supplier A has the materials that you want today, the possibility exists that they will not have it for you tomorrow. And so we've gone to great lengths internally to really ensure that if that were to happen, that we have a default supplier for those things. And then wherever possible, what we've done is and I think this is a lot more, quote, doable with innovator companies, is to ensure that as you're developing multiple products, that they're utilizing similar reagents or supplies, because at that point you have redundancy of function in terms of manufacturing. And it also obviously lessens the burden on your warehouse operations as well. All of these things feed in then to scale from a supply chain perspective. And I think it becomes a different conversation after the pandemic. But at present, I think the bulk of what we do is just keeping our head above water to ensure that we're able to create the immediate demand rather than the longer term best case scenario for Allo considerations or the like.
Yeah, sure. And Mike, I'd love to get your perspective on this, especially given your background. Like ensuring that supply chains for the can support product duration, and again to mitigate those delays, and what that partnership looks like. What's your take on that?
So let me just be really high level and then talk a little bit about how it relates to how we develop our products. So ideally, any supply for a process, if you can have everything be transactional suppliers, meaning lowest bidder wins and it's all the same stuff, then you reduce a lot of risk in your supply. The problem is I don't think we will never be there with cell therapy. But this is where risk assessment becomes important, right? What are the important supply components of your product? For example, serum, right? If we develop the product with a particular serum and we didn't explore other sources of that, you could be in a situation where you have to have a strategic agreement with that supplier, meaning your product its success in the marketplace depends on the success of that supplier itself. So, and also that takes rents it increases cost of goods because you're locked in with a particular supplier and they know it, right? So things like serum, media that can be very proprietary, sources of things like IL-2. There are multiple manufacturers of IL-2. Do they all work the same? When we develop our process, we should be thinking about this and exploring this. And this is, again, goes to what I'm talking about, post-approval changes, right? We have to be able to react to supply chain challenges. That, I think, is a key component of developing a cell therapy product, be it autologous or allogeneic. So my take on that is understand where those potential strategic suppliers lie, and then factor that into your product development plans so that you can get a few degrees of freedom there. Now, things like vector manufacturing, if you don't make your own vector, yeah, initially you're probably locked into one supplier because that's essentially its own BLA, right? Think about it there too. This is where your post-approval changes come in. Think about it long-term. You're supplying a market forever. Everything, strive to transactional suppliers. Use risk assessment to help you with that.
Beautiful. Okay. Mike, earlier, I want to make sure we're getting to patient scheduling, chain of identity, tracking, all of that. And let's, keeping scalability in mind, for that autologous one patient, one dose, how does patient scheduling fit into this? Because I know it's a critical component. So talk to us about that.
This is for me?
Yeah, Mike.
Yeah, sorry. I get glitches now and then. I said it before, right? So if we are supplying a market and it's on the patient's time, right? We have 28 days, let's say, to get from that apheresis to a product in that patient, dosing the patient. This relates to equipment utilization. If we can get to 70% utilization, meaning 70% of the time that equipment's making product, 30% of the time it's idle, waiting for product or under maintenance, then you're pretty golden, I think, in cell therapy. But what does that mean to cost of goods? If you're 50% versus 70% utilization, that's a 50% larger facility. More stations, if you will. Mm-hmm. So that's how I look at it. I don't really have much more to add to that, actually.
Sure, no problem. And before I want to make sure we give plenty of time to our Q&A from the audience. But before we do, I want to make sure that, John, if you have anything you'd like to add for patient scheduling while we're on that topic, please feel free.
No, I think we can move on. I think you did a really nice job covering the high points.
Good. Okay. All right. So we're going to formally move on to our Q&A from our audience because we have quite a few. So one, we want to discuss how are single-use materials disposed regarding environmental effects? And either one of you could take that question, but
I could take that one.
Feel free to share.
It's considered infectious, so you have to have them. It depends on your throughput in your facility, but waste inactivation becomes really important. So you have to have strategies to inactivate that waste. I recommend you qualify that so that we don't infect anybody. Remember, everything that we manipulate in cell therapy, especially autologous, we have to consider to be infectious.
Mm-hmm. Mm-hmm. Okay. John, there's a question specifically for you. What type of characterization do you think will become necessary for healthy donor starting materials?
Yeah. This is it's an area that's still under active discussions with the agency, but obviously, the big ones that that become quite important from a safety perspective are genomic sequencing of the donor cell, both pre and post genetic manipulation. And I know that like companies like Fate Therapeutics that are doing this, they're looking for genetic drift, if you will, over time to ensure that stability is maintained as the drug product, as we go over time. HLA background, I think, is obviously going to be important for matching purposes. And then in terms of just the actual functionality, the considerations that a lot of companies are looking at the moment, age is going to be one. I think earlier age cells are thought to be, and there's probably enough clinical data at this point to support the hypothesis that they're going to be more efficacious. The phenotype that you've heard me talk about a number of times, where T cell therapies are concerned, understanding the departmental subsets like stem cell memory and so forth. Carl June's team published a paper looking at in Nature earlier this year looking retrospectively at some of those T cell compartments and showed that CD4s are particularly important for sustained responses. And so you could imagine a scenario where companies want to ensure that that CD4 compartment is there as well. And then I think that we touched on the other ones are around expansion capacity, because without that you're not going to be able to achieve the numbers of cells that are needed for an allogeneic drug product. And then lastly, and potentially most importantly, is cytotoxic profiling. Understanding that, hey, if you've got something that can expand to 10 to the 14th in a batch, that cell still maintains the level of cytotoxicity that it needs to in order to achieve its downstream intention, and that is to, in this case, using the example I used, to kill cancer. But obviously, there are other applications there. But those are the big ones that I think are going to be the focus of innovator companies that are looking to derive healthy donor-derived allo treatments.
Good. Okay. Mike, I have a question for you. You were discussing serums earlier at the in our call, and so I wanted to ask you this question from the listener. How seriously should serum-free media solutions be considered to streamline supply chain?
Nirvana. I mean, there's a lot of issues with serum. Human serum or bovine serum certainly out, right? There was a time 5 or 6 years ago, bovine serum was being phased out, now gone, right? There's a lot of risk associated with serum. And if we can get a serum-free process, then the regulatory hurdles around safety could be addressed much better. Okay, so with bovine, of course, we have bovine encephalitis. You have similar issues with human serum. Also, the variability of human serum as well. The source of human serum. If we look at it purely from a supply chain perspective, that's one of those strategic suppliers you just don't want to have to deal with. So if I could have a process where I get, if it's an engineered T cell, right, where I get vector for the cost of a monoclonal dose and no serum and media that's not proprietary, that would be great. For a TIL process, we use feeder cells and TILs. Get rid of the feeder cells. There are major safety issues with that. Same thing, get rid of serum. I would have a more valuable product, and my cost of goods would be lower. Even if the result is a couple extra days of processing to get to dose.
Okay. I want to get to our next one. We have so many. Have you seen any input from regulatory agencies regarding capacity modeling protocols and requirements for submission data? Mike, we'll start with you with this one.
This is one of the main things that, in monoclonals, my background is in validation. Okay, so I've been validating things since the early 90s. And in monoclonals, we look at capacity of a particular piece of equipment. That bioreactor can have X number of liters, or that CIP skid, clean in place skid, can clean or can dose cleaning agent in a specific capacity. When it comes to autologous cell therapies, you have to look at your facility as the system. And really exercising the throughput, I think, is a core component of process validation for these types of products. Remembering, given the analogy I gave, it's not the end of the world, is what I'm trying to say. Given the analogy I gave about the salt solution, the days of, back in 1993, when I did my first process validation for a monoclonal, it was 3 lots, right? And then the 2011 guidance came out, the life cycle guidance. Then we had the EMA, the Annex 15 came out. And Annex 15 said something like, notwithstanding the traditional 3 lots, you have to have statistical significance, right? This actually marries quite nicely with throughput validation for a cell therapy facility, because I will guarantee you're not going to validate a cell therapy product with 3 lots. It's going to need a lot of lots. I've heard upwards of 30 lots. maybe 60 lots. You don't know. Especially now, if you add vector, you need multiple degrees of freedom there, and you could have 8, 10, 12 lots for that process validation. So you think of that throughput validation in terms of what's required for the process validation, but you do have to address throughput.
Okay. John, I have a question for you. For developing in-house versus outsourcing media, what are the major factors that would help you measure cost benefit for each scenario?
Yeah, this is an area that we're actually looking into quite a bit at the moment. The answer is actually fairly straightforward. It's looking at the considerations that I actually just described for understanding healthy donor-derived cell selection. On the back end of that, you heard me talk about functionality and phenotype. These are going to be the output by which you measure the success of your media considerations. So if you start with the end, and that again is the efficacy and phenotype, at that point, it's just a matter of iterating your in-house media in such a way that in the end, you can achieve levels of cytotoxicity that are commensurate with your outsourced media, and maintenance of that phenotypic profile that you desire. So, and then it's just a cost game, right? You look at what it costs you to develop that in-house media, not just develop, but obviously engineer and generate over the long term versus what you're paying for the outsource. And then it's a fairly simplistic math equation. If you're getting the similar phenotype at a reduced cost, then you go with the internal derived media and vice versa.
Good. Okay. Well, everyone, we have come to the end of our hour, and that officially wraps up our Cell and Gene Live event. Thanks so much again to Michael Blackton and John Lee for their time and insight. Gentlemen, this was wonderful. Thank you so much.
You're very welcome.
The on-demand version of this event will be live on Cell and Gene soon, and be sure to bookmark cellandgene.com to access our always informative content. And we will see you next time.